It's my great pleasure to introduce the keynote speaker of today, Miháli Pósfai from the University of Pannonia. Mihály has received a master in geology from the university in Budapest and has visited Arizona State University as a postdoctoral fellow, the University of Cambridge, Swedish Institute in the Department of Inorganic Chemistry, and is for a long time a professor at the University of Pannonia, very well known for environmentology and nanogeoscience using specifically transmission electron microscopy. So I'm very much looking forward to his presentation from dust to bugs, transmission electron microscopy as a versatile tool in environmental mineralogy as our keynote speaker in this exciting meeting. Mihály, by the way, would like everyone to mute their microphones if possible. And Mihály, the floor is yours. You're muted right now, by the way. We should be able to hear you now.
Okay.
Yeah, perfect.
You still see my slide, right?
Yes, we do.
Okay. Okay. So thank you for the introduction and good afternoon to everyone. I hope you have had a wonderful 2 days of workshop by now. So I'm going to talk about environmental mineralogy problems. We are based in Hungary in the city called Veszprém, and we call ourselves the Environmental Mineralogy Research Group because our topics are quite diverse as is listed here. I'm not going to talk about everything today. The first part of the talk will be about carbonate precipitation in lakes and phosphorus issues. And in the second part, I'm going to talk a little bit about magnetotactic bacteria. And then if we still have time, then about the dust, the atmospheric aerosol particles. Before I go on to these topics, I'd like to summarize what I think why transmission electron microscopy is such a great technique. It's because simultaneously you can have all this information with the same instrument about the same sample. I'm sure you are all familiar with this now, but I'd like to emphasize that it's not just images, and images sometimes give you information about composition when you do elemental mapping, also about structure, but there is also electron diffraction and also electron tomography for the three-dimensional shapes of nano objects. Actually, this group of techniques is available in our lab. We have a Talos electron microscope, so we can do all of this. And there are some more advanced techniques like electron holography or environmental TEM or cryo-TEM that you can do somewhere else. I'm not sure if you covered this in the last 2 days, but of course, all of this is very useful. What I'd like to emphasize that TEM data are often images and beyond the scientific content of the image, it also has great power in conveying your message. On the other hand, you also have to be cautious because images are easy to be misinterpreted because everyone thinks he or she understands what's in an image. And I had this experience with people who don't use electron microscopy all the time, that they tend to see things in images that they want to see in them. And that is very dangerous sometimes, so you have to be careful with that. The worst case is when someone comes to our lab and tells me that, please take a few pictures because it will look good in the paper. So actually this slide is a kind of a summary. I should have put it at the end. So the rest of the talk is just illustration of what was in this slide. So the first topic is carbonate precipitation and lake sediments. This is Lake Balaton, which is very close to us. And this is very important in Hungary because Hungary is a landlocked nation. And this large lake is 70 kilometers long, and it's both culturally and economically very important. So we wanted to see how carbonates form in it. and phosphorus is certainly also an important issue. Sometimes, especially in the spring, the water becomes milky, and that's because of very intense photosynthesis of algae. And when the algae photosynthesize, they change the pH. The pH gets higher, and then calcite starts to precipitate, or carbonate starts to precipitate. So these are called whitening events. And it seems like a simple process, but it's not as simple as that, because for example, heterogeneous surfaces play a role in it. There is a lot of literature about this, that the surfaces of algae can help as nucleation sites for calcite. So we wanted to see what's the relationship between algae and carbonates, and which carbonate polymers nucleate, and how do they form. So we had a chance to sample a big bloom of algae. And in this algae bloom, we found many microorganisms. For example, this is a cyanobacterium train, cells that form filaments. And next to these cyanobacteria, you see these little dots. Those are all amorphous calcium carbonate minerals. Here again, this is made of silica. And next to this silica, which is probably a spicule of a sponge, you can again see the amorphous calcium carbonate particles. And these are, as I said, amorphous, but they produce typically these two rings in the electron diffraction pattern, so it's easy to identify them. And it's interesting that their composition is not homogeneous, because they have a magnesium-rich rim and also contain some sulfur. So actually, we think that this is the first precipitate in the lake, but we only see this next to algae. And what happens to it later? Well, actually, half a meter above the level where most of the algae are, we see something different. We see some spindles of carbonates. Sometimes they are close to bacteria, sometimes they are just by themselves. And they can be either aragonite or calcite. I didn't say that this lake is magnesium rich, so this is why you have aragonite. Again, they have this heterogeneous composition with a magnesium-rich rim, but you can also see something strange in it, especially in the elemental maps. The red is silicon, and you see that there is silicon also here and around the rim. So there's something, a silicate in these particles. And what is it? Actually, if we blow up this part of this spindle, we see layers of a clay mineral, smectite. So just a few atomic layers in the middle and also on the edge. So probably this carbonate spindle may have formed on the floating flakes of smectite. What do we see when we go away from the algae? For example, we look at the sediment, or we collect some suspended particles. We see different things. We see magnesium-bearing calcite, which looks like an aggregate, but in fact it's a single crystal. It's elongated. This one and that one, they all look quite the same. And actually, this is the main material of the sediment of this lake. You have 6 meter thick mud that it consists of this magnesium-bearing calcite. So we think that this is the end product of the calcite precipitation. And what is interesting that, again, if you look at this at high magnification, this is the calcite, the blue part, and you see the red, the silicon distribution on the surface and also inside. And when you look at high resolution, you see the just 5 layers of this smectite clay mineral. So from this, we had to ask this question, whether the smectite acts as a nucleation surface for calcite. And to understand what's happening, we did 2 things. We did calculations, molecular dynamics calculations, where we took layers of smectite and put the lake water between the 2 layers and checked what is happening here. Well, actually, you see there are some clusters of ions here forming, so-called pre-nucleation clusters maybe, and some of them get caught on the smectite surface. And we did the same experiment with kaolinite and also with graphene. And the result was that smectite is really efficiently capturing these particles, whereas graphene and kaolinite are not that good at doing it. But this is actually just 30 nanoseconds, so this is still not even ACC. It's just ion clusters getting attached to smectite. But we think this is the process what is happening. The other approach that we took was that we did some lab experiments where we had a carbonate-bearing solution and titrated calcium-bearing solution into it. So on the horizontal axis, you see an increase in the calcium concentration as it is added to the solution. And on the vertical axis, you see the free calcium activity in the solution. And as calcium is added, of course, calcium activity increases. And then when it drops, that indicates the point where nucleation happens, where the solid phase starts to form. And the blue curve represents the homogeneous solution when there is no clay mineral. And when you add clay mineral into the system, then what you see is that nucleation happens much earlier. So you don't have to add that much calcium. You don't have to have such large supersaturation. The smectite really seems to aid in the formation of calcite. And it's not only that, but this clay mineral also has an effect on the polymorph that forms. Because if we have a homogeneous solution, first we have ACC, amorphous calcium carbonate, but that's hard to catch. And the first phase that could produce a diffraction pattern in our system is the calcium carbonate polymorph called vaterite. On the other hand, if you have a smectite flakes in the solution, then immediately you jump to calcite. So somehow the smectite flakes make calcite. This was in a solution that contained only calcium, but in the lake you had also magnesium. So when you try the same thing with a magnesium-bearing solution, the result is slightly different. You don't have calcite, but you have aragonite spindles. As you see here, the clay didn't have such a large effect because even with the presence of smectite, we ended up having these aragonite spindles with some clay flakes inside the spindles. Actually, this looks very, very similar to the objects that we saw next to the algae in the algal bloom. So far, we didn't really get that kind of magnesium-bearing calcite that forms the sediment. However, if we age these aragonite spindles with the smectite in a solution for a couple of months, then we end up with some very interesting objects. These are some composite particles that have a core. You see this euhedral calcite particle with some magnesium in it. So blue is calcium. And that is surrounded by filamentous structure. And the filaments are, each of these little filaments are made of nanocrystals of protodolomite, which is a magnesium-rich phase. So this has two parts, a core and protodolomite around it. And the third part is the smectite. Here you see the red, again, inside and on the edge. And on this tomographic reconstruction, you can see that in the middle where you have this calcium core, the red stuff is again the clay mineral here. And clay mineral also occurs in the outside. So how did we come here from the aragonite spindles? We believe that there are some dissolution and reprecipitation processes. And actually, the clay mineral's role might be to provide some space, some compartmentalized space, and also a nucleation surface for that. So when you dissolve a phase that is not as stable as another phase, and you have some extra supersaturated solution in the closed space provided by the clay, then you can have crystallization of a dolomite-like phase. But do we see this protodolomite in the lake? The answer is yes, but before we go there, I'd like to explain what I mean by protodolomite, because it's not that easy So everyone knows about calcite and dolomite. Difference is that in dolomite you have as much calcium as magnesium, and in alternating layers you have only calcium and only magnesium in an ordered fashion. And between calcite and dolomite there are lots of phases that go by different names. I just want to point out that this protodolomite can be considered something that it has a composition close to that of dolomite, but it's not completely ordered, but it's kind of weakly ordered. But what does that mean exactly? Well, protodolomite was described really a long time, more than 50 years ago from Lake Balaton. And we just wanted to check if it still forms in the lake, and what is this weak ordering physically, what does it mean? Well, if you collect some particles from the water, suspended matter, that this is what you see. There are some diatom fragments, and all these numbered particles are carbonates. And most of these carbonates are just magnesium-bearing calcite. Those are the blue ones. And usually they have this ratio in them. But sometimes you can fish out these yellow ones in which the magnesium-calcium ratio is larger than half. And if you take electron diffraction patterns from these particles, then you see some extra reflections marked by these arrowed rows that can only come in dolomite. So that already suggests that calcium and magnesium are ordered to some extent. And some of these particles have very interesting compositions. Firstly, you should notice that, again, we have this red clay mineral all around it, and even it shines through, so it's sometimes in the middle as well. And also that the blue and the yellow, the calcium and the magnesium are zoned. There is a high magnesium core, then a low magnesium zone, then again a high magnesium zone. And if you take electron diffraction patterns from the yellow part, from the high magnesium part, then you always see these extra reflections, the rows of extra reflections marked by red arrows. Sometimes they are stronger, sometimes they are weaker. So electron diffraction gives you an idea of the bulk structure, because we have a pretty large aperture, and you take a diffraction pattern from one part of this grain. So on average, we see some sort of ordering, but we don't know how it is exactly on the nanoscale. Well, to see that, we took some high-angle annular dark field images where each of these dots represent a cation column in the structure. And you see some double periodicity here in this domain, whereas here you don't see that double periodicity. So there are some ordered domains and less ordered domains. The next micrograph, there is a more detailed analysis of the whole thing. Like here in the yellow square, and this is filtered here and averaged at the bottom, you see that every second row is bright and every first row is less bright. Whereas in the red square, each spot is the same. Well, actually, in STEM high-angle annular dark field images, the contrast is proportional to atomic number squared. You probably learned about this in the workshop. So that means that calcium-rich columns are bright and magnesium-rich columns are less bright. So from this, we would think that this is just ordered dolomite. However, from the intensity, you can actually calculate the occupancy of these individual columns. And we ended up with this composition. So in this ordered part, you have the bright columns have twice as much calcium as magnesium, and the less bright spots have twice as much magnesium than calcium. And in this part, you have approximately the same amount of magnesium and calcium in each column. So protodolomite can be characterized by these few nanometer large domains. This is 5 nanometer, and the domains are approximately the same size. And we think that we have this in the lake. So this is a summary of the carbonates in the lake. So next to algae, we see magnesium-bearing amorphous calcium carbonate spheres that transform somehow into these spindle-shaped particles. Well, there is a very rich literature. I don't didn't put here the citations on how ACC converts to either aragonite or calcite. But in this case, in almost all of them, we find smectite clays. And that's because in this shallow lake, I forgot to say that this large lake is very shallow, only 3.5 meters on average. So even the slightest breeze can stir up the sediment. And these large nanometer scale flakes almost always float around in the water. And whenever the ACC spheres or just ion clusters find smectite, they attach to it. And once they are attached, then they can crystallize into calcite or aragonite. When there is smectite, it's preferably calcite. We don't exactly know what's happening here, but since we never observed these things only next to algae, we think it happens very fast, that it crystallizes into this aggregate-looking single crystal calcite. And protodolomite is a minor phase in the lake. Now, we actually recreated the whole thing in the laboratory, because if you do the reaction rapidly, then you end up having the same magnesium ACC. However, if you have only magnesium in the solution, then you skip this part, and on smectite, you immediately have calcite. A slightly slower procedure was the titration, which could be matched with this spindle things. We ended up with the same spindles in a magnesium-bearing solution. And again, if there was only calcium in the solution, we jumped immediately to calcite. And here, the correspondence between lake and experiment is not perfect because we couldn't make these magnesium-bearing calcite yet, but we ended up having this protodolomite with a calcite core. So this is the takeaway message that smectite helps a lot in making calcite. So, and that's not a big wonder because the surface of smectite can probably template a calcite formation, but it doesn't happen in a way that we first thought, that individual calcium ions would arrive on the surface and make a crystal immediately. What we think is happening, that the nuclei form in the solution and they get just fished out by the smectite surface. So probably it's ACC or just ion clusters that arrive on this surface. And then when the surface is covered, then they are organized into calcite. And of course, magnesium makes the whole process slower. That has also been known. And we described protodolomite structure that has nanoscale domains of ordered magnesium-calcium distributions, but it's not perfect ordering as in dolomite. Well, then we still stay with the lake, but now I'm going to talk a little bit about phosphorus. I have to admit that I'm a little bit concerned to talk about phosphorus here in Utrecht, because there are many people here who know a lot more about phosphorus than I do. And also, all our results are very preliminary, not even published. No screenshots, please. So anyway, there was a large bloom of algae 2.5 years ago. It was really ugly. So on the satellite image, you see there's a huge amount of chlorophyll forming. And the funny thing about this was that at first sight, nothing special happened. So no one expected this. And some people calculated that 60 tons of phosphorus was needed for this large bloom. And this is supposed to be a very nice, clean lake in which people swim all the time. So it's not very pleasant when something like this happens. And the inflows are all monitored, and there was no extra phosphorus coming in. So it could only come from the sediment. But why would it come from the sediment? Well, it was turns out there was some intermittent stratification periods, which is rare in this shallow lake, that could trigger the phosphorus release, and that would point to iron oxide minerals. And we know that phosphorus is mainly bound by carbonate and iron oxide minerals in this lake. But how do we know that? Well, typically from sequential extraction procedures. There are many, many ways of doing this. People use these reagents to get phosphorus out of sediment, and then they use these terms like loosely bound, Fe bound, calcium bound. And actually, this paper is a review of all these techniques that are being used. But when you think about it strictly mineralogically, this is not very satisfactory to say that this is calcium bound and organic bound. So we wanted to see if we can find visible phosphorus. By visible, I mean that it's visible by TEM. And can we see it on carbonates and iron oxides? And can we experiment with it and see when it gets released? Well, first we looked at the carbonates. Again, the dark blue is calcium. So this is a calcite particle. Here you have the smectite on it. And the light blue is phosphorus. And you tend to see here that just under the smectite, there is a stronger hue of light blue. And also in the spectrum, there seems to be a little peak of phosphorus. So at first glance, okay, we have some extra phosphorus here. However, you have to be aware of some artifacts in EDS spectra. And actually, if you have a lot of calcium around, and if you want to analyze phosphorus on calcite, you are definitely having a lot of calcium, then the escape peak of calcium, so calcium peak minus the silicon peak, falls here just next to phosphorus, and the two are overlapping. So this peak is an artifact coming because of the silicon detector and makes the analysis of true phosphorus a little bit more difficult. So we think that our detection limit for phosphorus in the presence of calcium is worse than it would be otherwise. Maybe 0.2 atom percent is needed. And the phosphorus concentration is just an order of magnitude lower than that. So we think that the distribution of phosphorus might be uniform on the calcite, and it's below the detection limit of EDS. So this, although this talk is supposed to be showing the advantages of TEM, there are some limits to even to TEM in this case. And all these element maps can be really deceptive, because if you just subtract the background from here and make an element map using these peaks, then you will end up with some extra phosphorus here, where there is a lot of calcium. So on calcite, we didn't really see it. How about iron oxides? In the first place, in this lake, there is not too much iron oxide at all, but there is one part, exactly the part where the bloom occurred, where we found ferrihydrite. So these green dots are all ferrihydrite particles, and surprise, they are on smectite clay. Even the ferrihydrite is on smectite. And we don't really see phosphorus, not much. Sometimes we do a little bit, but it's not what we expected, and it's not as much as we end up with in experiments. So no luck again. Even with iron oxide, we don't really see the phosphorus. However, we did see some visible phosphorus in the lake sediment. Again, the dark blue is calcite, red is clay, and between the calcite particles and clay particles, in some places, not very often, we see these phosphorus-bearing granules. And they have a fairly specific composition, and they are amorphous. And then we started to wonder about their origins. Is it inorganic? Is it biogenic? And it's tempting to think it's biogenic, because these are some cyanobacteria, and they have these polyphosphate granules. So microorganisms and all organisms tend to make these polyphosphate granules because they use it as a storage of phosphorus and also for metabolic reasons. And they look similar in size and composition as the phosphorus grains in the sediment. So what we are doing now is that we try to take samples when the algae bloom. And then in this algal bloom, we have all kinds of features. The cyanobacteria, then there is this probably a picocyanobacterium, I call it emoji. And there is this stamp-like cells, and they are all full of these polyphosphate granules. And if you analyze them using this STEM EDS elemental maps, then they tend to cluster. So what can we analyze in them? Of course, we cannot analyze really the carbon and the oxygen, but we can analyze the alkali and alkaline earth metals. And based on them, there are different clusters of compositions. And actually, the ones in the sediment fall somewhere here, so might even be in among these. But as I said, this is just very preliminary, and of course, we are still working on it. And although I wouldn't like to have a large bloom of algae in my lake next summer, if it happens, then we are going to sample it and check the phosphorus movement. So that was the first part. Now we go to some bugs, magnetotactic bacteria. I know that you saw already some examples of magnetotactic bacteria yesterday, so you know what they are. And actually, it's very easy to see them with your own eyes if you collect some mud and you put a magnet with the right pole next to the sediment water interface and leave it there for half an hour. And then you take a droplet with a pipette from here and then put it under the optical microscope in a droplet and put a magnet next to it. Then you will see them. And you will see something like this. This is the edge of the droplet, and these are all magnetotactic bacteria. If you turn the magnet around, then they will just race away. They turn again the magnet, they come back. So that's what they do. That they follow the magnetic field exerted by the magnet, because the magnet has a stronger magnetic field than the Earth. So the bacteria can be directed by the magnetic field. And how do they do it? Because they have these magnetic crystals in the cell, and these magnetic crystals tend to be magnetite. This one is a nice twinned crystal, as you can see here in the Fourier transform as well. And on an EDS map, you can see that there is extra phosphorus around them, and that's because each of these magnetite nanocrystals is surrounded by a phospholipid bilayer membrane. So the membrane and inside the particle together are called magnetosomes. They are little organelles in these cells of bacteria. So why are we interested in magnetotactic bacteria? Well, I will just show two examples. First, these are very interesting objects because they make magnetite or magnetic nanocrystals in different arrangements, so they can be used as models to understand magnetism on the nanoscale. And second, they are the simplest organisms that make biominerals. So by studying magnetotactic bacteria, you can learn a lot about biomineralization. And by learning from the bacteria, you can use that information in synthesizing some magnetic nanostructures. Well, actually, these bacteria each strain makes magnetic crystals with specific shapes. So some make perfect octahedra, which is not so surprising because that's the equilibrium morphology of magnetite. Some others make cubo-octahedra. That's also okay. This is already rather strange because you have these elongated barrel-shaped particles, and this is really weird. Some bacteria make these bullet-shaped particles. It's hard to make a model like that. So that's obviously, this is under genetic control, how they make this. And we are interested in which factors affect and how the magnetic nanostructures of interacting magnetic nanoparticles. So what are these factors that are of interest? The first is the domain size. So how large are these particles? Where for magnetite particles, if the particle is between about 30 and 120 nanometer, then you have magnetic single domain. If it's smaller than that, it's non-magnetic. If it's larger, it will be multi-domain. So the bacteria know that they should make magnetic single domain particles if they want to use it for alignment, and they do it. So there's not much question about that. They make single domain magnetite. But there are some other factors as well. For example, crystalline orientation, because in magnetite, if you magnetize the crystal, it's easiest magnetized along 111. The other directions are not as good. So this is called the magnetocrystalline anisotropy. particle shape is also interesting, because if you have elongated particles, that might force the magnetic field inside parallel to the elongation. And the last one is the effect of chain configuration, the interaction of particles. And all these effects are best studied in particles that are elongated. And fortunately, some bacteria make particles elongated along 111, some others along 100, and again, some others along 110. I'm going to show you examples for 100 because in this case, well, this one is not so interesting. I hope you see my cursor, because if it's elongated along 111, that means that the particle elongation and the magnetocrystalline easy axis coincide, so we don't know which effect is stronger. But in this case, 111 is somewhere here, so that would be the magnetocrystalline easy axis, and that's the elongation axis. But how do we measure the magnetic induction in these particles? Well, there is an advanced electron microscopy technique called electron holography in the TEM. As I said, we don't have this sort of hardware, and not just hardware, but also the knowledge to do it. But we collaborate with people in Cambridge and in Jülich, most of all with Rafał Dunin-Borkowski. Well, actually, this off-axis electron holography was invented by Denis Gabor, who happens to be Hungarian, but he worked in the UK and got the Nobel Prize for inventing holography. And this is what holography is about, that part of the beam goes through the sample and the other part of the electron beam goes through vacuum. And then there is a positively charged wire here that makes the two beams overlap. And as they overlap, they interfere with one another and make the hologram. And hologram looks like that. These are the magnetosomes, and you see these little fringes, and they change direction as they go through the particles. So that's a phase change. And this phase change can be described by this equation. So the phase shift in this picture depends on two things. One is the mean inner potential. That's just the amount of material that's inside. But the other one is the magnetic induction that is perpendicular to the electron beam direction. And there are some experimental and also some other ways to get rid of this part and measure only this part. So that's the result is best seen in images like this one, where you have this chain of magnetosomes in a bacterium. And this is the magnetic induction map from electron holography. And what you see here is one thing is the color. So most of them are green. Green just means that the magnetic field, magnetic induction is in this direction, or slightly yellow when the magnetic induction is in this direction. And also these fringes show how the magnetic induction changes as you go across the particles. And you see that they have a net magnetic moment in this direction. And also you can calculate the projected magnetization inside these particles. So it shows how these particles are magnets. And you see that the bacterium made a beautiful compass needle. So it's just a nice magnetic dipole. So how do we study the effects that I mentioned? Well, this is an isolated crystal that is elongated along 100. So as I said, the 111, the magnetic easy axis would be in this direction. But as you see, all the magnetic induction lines are parallel to the elongation of the particle. So that means that the particle elongation is more important than the magnetic crystalline anisotropy in the case of magnetite, because that has a stronger effect on magnetization. And by the way, this electron microscopy, electron holography is a quantitative technique. So we can measure the magnetic moment, which is 0.5 Tesla, what it should be for magnetite actually. But these experiments were done by András Kovács and Rafał Dunin-Borkowski in Jülich Research Center. If we have these particles in a chain, it's a quite disordered chain, then we see some interesting things here. And in some cases, the magnetic induction is again parallel to the elongation of the particle. But if you look at this particle sandwiched between two others, then you see that all these field lines go across the particle. And that's because the magnetic field of these neighbors forces the magnetic induction to be parallel to the basically the chain axis. So from this, we can gather that these interparticle interactions override the effects of particle shape. So the most important is in such a system, the arrangement of particles. The second most important is the shape of the particles. And third is the magnetocrystalline anisotropy. Well, the other topic about magnetotactic bacteria is the genetic information on biomineralization. This work was done in Dirk Schüller's lab. He's now at the University of Bayreuth. And they are biologists working on magnetotactic bacteria, and they identified lots of special proteins. All these MEM things are special proteins that are inside the magnetosome membrane. And these proteins are associated with magnetite formation. So what is the sequence of events leading to magnetite magnetosomes? First, the inner membrane of the cell invaginates like that and forms a vesicle. There are some proteins that direct this process. Once you have a vesicle, then you have some ion channels through which iron ions are taken up from the environment. So that's regulated again by some proteins. And once there is supersaturation of iron here, then magnetite nucleates. And there are some proteins, among them MEMI, that are thought to enhance the nucleation of magnetite. And then the magnetite grows, and even the shape is controlled by some proteins. And finally, all these magnetosomes are attached to this filament marked K by this MEMJ protein. So that's the somewhat simplistic view that each of these proteins has a function in making the magnetite. How do we know that? Because these biologists made some mutants of these bacteria. They deleted some genes that control the formation of these proteins. And I showed just one example. That's the most spectacular one. Like this was the original bacterium, and this is the mutant. And this mutant differs from that one in just one single gene. This MEMJ was deleted. So it didn't have this protein that attaches the particles to the filament. And all the particles are the same size, the same composition. Even the number of particles per cell is the same. They just cannot be in chains because this protein is lacking. So this is how we know the information about what these proteins are doing. But in most cases, it's not so simple because they have all sorts of synergistic effects. So based on this information, we devised some scheme to make magnetic nanofibers. What we thought was that we took one bacterium, a Salmonella, which is very good at making these filaments flagella. That's the organ for motility of these bacteria. And we wanted to modify these flagella to make them magnetic. And these flagella are made of this flagellin protein. And this protein has many, many copies of the same protein. And this protein has this domain, the D3, which is outside here. Every 9 nanometers, you have this D3 domain. And this can be quite easily replaced. And we replaced it with a polypeptide from the magnetotactic bacterium. So with MEMI, for example. So we thought that MEMI nucleates magnetite, and we put that in here and made mutant bacteria. So all these colored flagella are the mutant flagella that were modified to make magnetite. And then we put these mutant cells, actually we cut off their tails, and we just did the tails. And we had 2 approaches. One that was, these were put into suspension of magnetite nanoparticles. By that, we wanted to see if particles get attached to these modified filaments. And the other was that we put it into iron-bearing solution, where we thought that the magnetite would form on the filaments. And what did we end up with? Well, it's a lot of work, and it's we were happy to have beautiful mutant filaments. These are all the mutants, because sometimes if you modify biologically something, maybe the bacterium would not grow a flagellum at all, but these worked. And then indeed magnetite particles attach to these filaments. So this is nice. However, you see that the coverage is not perfect. In some places you have magnetite, in others you don't. And the interesting thing was that no matter if we use the pre-made magnetite nanoparticles, as in this case, or we put the filaments into solution, we ended up having the same thing. So even with the nucleation experiment in the solution, we didn't get a complete coverage of these filaments. So more than 20 distinct mutants were made, and we found that particle binding and nucleation from solution are not that different, probably because nucleation also happens in the solution, and then these clusters or first maybe amorphous particles get attached to the filaments. So that's why it's not so different. And it puts the whole biomineralization by magnetotecting bacteria in a different light, because we found that magnetite nanoparticles attach to almost everything. Actually, in this paper, for example, magnetite particles were attached to eggshell membrane. So maybe all this work with the mutants was in vain. And now I think that maybe in magnetotecting bacteria, the special protein in this vesicle, the function of it is not to nucleate magnetite, but to prevent nucleation so that only one crystal would form. And there was one more interesting result from this experiment, that the shape of the magnetite particle has an effect, because if we experimented with octahedra, then that was more specific. But if we experimented with cubes, which have higher surface energy, then it attached to everything, even to some controls. So particle shape is important in this attachment thing. And once we made these filaments, we also tried them to make it useful. And actually, if you have a suspension with this magnetite covered filaments, and then you turn on the magnetic field, then the viscosity increases a lot. Whereas in the other cases, if you have just filaments or just magnetite, there is no such effect. So maybe it can be used for some technological application. Well, I think we started a little bit late, so maybe I still have 5 minutes to talk about atmospheric particles. But if you think it was enough, then please.
No, please go ahead.
Okay. So that's a totally different topic, but it also uses a lot of electron microscopy. So we study particles in the atmosphere, aerosol particles, because they are important for global climate and also regionally and locally, as you all, I'm sure you all have noticed that. And this is a huge field. It's much larger than the normal mineralogy field in science. And mostly people, what they are doing, they do physical measurements and they do chemistry of the bulk aerosol, and they do models. And TEM has an interesting niche in this area, because I think it's very important to know what the individual particles are doing. So the atmosphere is not like a big beaker, because the particles are there individually. So each particle is a beaker, and chemical reactions happen in them. And for individual particle studies, mostly mass spectrometry is used, but TEM has been applied in the last 50 years, but it can provide some very interesting results, I think. So my first example is so-called biomass burning. That's a fire in Zambia. And this is not just a local issue, because if you look at this satellite image that was taken at the same time when this fire was burning, you see clouds, those are the white things, and these gray areas. The gray is all smoke from biomass burning. So it has a regional effect, and it also has a global effect on climate. And that's a long story. That was more than 20 years ago. We participated in a campaign where particles were collected from this smoke. And there are different types of carbonaceous particles. First one is soot. Well, the soot, that's very common. It's also called black carbon, because it's the most important absorbing particle in the atmosphere. And this is what comes out from the tailpipe of cars, or if you burn a fire or whatever. So it has these little spheres. They form these grape-like aggregates. But if you look at high resolution, you see these little layers. And these are all graphene-like layers wrapped around one another like in an onion. So that's typical for soot. It has this interesting nanostructure. And of course, this is one of the main components of any fire, and that's black stuff. But in this African biomass smoke, we found another particle which had larger spheres. It was also made of carbon and a tiny bit of oxygen, but it had no internal structure. It was totally amorphous, and it didn't form these grape-like chains. And we named this particle tarball. Actually, that was my that time Professor Peter Bisek's idea, and I just couldn't understand why he calls them tarballs, because we didn't know if it's tar or not. And it turned out to be quite interesting because people started to find them in every place of every corner of the world. But on the other hand, we didn't know what is the optical properties and what are the chemical properties, because if you burn a whole forest, then these particles don't come just by themselves, but there's a lot of soot and other thing next to them. But fortunately, in our university, there's an atmospheric chemistry group, and they came up with an idea to make tarballs in the lab, and without burning things, without flames. And that was just heating dry wood. And in wood, you have these channels, and by heating wood, you eject the tar from the wood. And then if you condense it, and the condensate is again heated, and you make aerosol particles, then you can end up with beautiful tarballs that look just like the real ones. And actually, even the compositions were similar, and all the sizes and the structureless features. So once we have the tarballs, then we can study their properties. Why is that so important? I mentioned that soot is the main absorber, but there's a term called brown carbon, which is not soot, but it's carbonaceous and brown, and that is thought to be responsible for 20% of global aerosol light absorption. And so we wanted to know what are the optical properties, and once we had a clean sample, then all these numbers could be measured, and that means that indeed it's in this range of brown carbon. So this is soot, this is transparent, and tarballs are somewhere here. And in addition, they also absorb in the red and infrared, so they have an additional contribution to a warming effect of our climate. And then we also characterize the compositions of these particles. Actually, I selected this example because this was a particle type that was identified using TEM, and now the properties of this particle type are, I think, included in all these global climate models that tell you whether it gets 2 degrees or 3 degrees higher next year. Just very briefly, another atmospheric topic, magnetic particles in urban air. Well, a few years ago, there was this paper by Barbara Meyer about magnetite nanoparticles in the human brain. Well, actually, in human brain, magnetic particles have been found much earlier, but in this paper, the conclusion was that these must have come from outside. So somehow they managed to get through the blood-brain barrier, and they got into the brain from the air. So might be some pollution particles. And the source of these particles is traffic in the cities. So we devised a very simple collection device. So it's a funnel, and there's a magnet here, and the TEM grid is placed here. And we sampled tailpipe emissions and also particles emitted by brakes. And what we found was that magnetite and hematite particles, micron-sized particles, but all of these are made up of just 10 nanometer nanocrystals. So that's why you get rings in the electron diffraction pattern. And this is a ring of showing the mixture of magnetite and hematite. But the interesting thing is that from the engine emissions, we get mainly magnetite, whereas from the brakes, we got mainly hematite and metallic iron. And we also collected particles with a personal sampler in a car cabin while driving. So while you are driving on the freeway, you inhale both magnetite and hematite, not surprisingly, because you get it from the emissions and also from the brakes. But in ambient urban air, it's mostly magnetite. So we think that this is the more important source at present, at least in the city of Veszprém. Another interesting thing is when you look at these particles at high resolution, the first thing you notice that these are the small magnetite nanoparticles. They are all in random orientations, and they are always associated with carbon. Sometimes the carbon is just amorphous on them. Sometimes, in some places, it looks like soot, so there are some layers. And in some places, it's more ordered. It's almost like a nanotube. So this is the way how nanotubes, carbon nanotubes are made. So there are some catalyst iron oxide particles, but this is happening in the engine or when the flue gas or the exhaust gas leaves the engine. So we think that these probably come from a high temperature source, the engine of the car. So that was the takeaway message, that the majority of magnetic nanoparticles appear to come from internal combustion engines. But of course, the more we use the electric vehicles, this might change, and the brakes also need a lot of attention. Well, these were the examples. This is my summary slide. I wanted to make it more elaborate, but I didn't know how to summarize carbonates in lakes and phosphorus and magnetite in the urban air. So that's the conclusion. And I wish to acknowledge the help of many people. From these many names, I would like to point out that Zsombor Molnár did most of the work on the carbonates. I'm not just saying it because he's listening now, but he really did a great work. And Peter Pecker did the high resolution dark field imaging and all the electron tomography measurements. And these two guys are responsible for the electron holography, and the air chemistry group, Adam Todt and András Hoffer, made the tarball experiments. And I'd like to thank very much for the consortium for this invitation.